आप कनेक्टेड है, यू आर कनेक्टेड सर। अभी सर ऑडियो इन नहीं है सर, हो गया सर, अब हो गया। ओके सर।
Let me share. Where do I share? आपने मुझे वो भी देना है। sharing rights are still not there, Vikas. Vikas, I am still trying to share the presentation. You have to bear with me, huh?
Sir, yes, sir. You're audible, sir. And
Yeah,
sorry,
no,
the
no
network
problem.
activities.
But just hold on. I'm just trying to share the presentation, huh?
Okay, sir.
Can you see my presentation?
Yes, sir, I can see. Sir, can we get the recording of this session? Yeah, thank you, sir. Thanks a lot. So that I can share it with the participants who were not able to join this session.
All right. No problem.
Thank you, sir. So I guess there are 35 participants till now, and should we start?
Just a minute.
Yeah.
Just wanted to see if the video is on or not. Okay.
So, dear participant, allow me to introduce very learned delegate, Dr. Alok Adholeya, sir. He is having over past 35 years significant experience and expertise in the area of translational research and reaching out to the millions of common people worldwide through his developed technologies, which include a new biological organism for soil fertility and crop productivity enhancement, known as mycorrhizal bioremediation of industrial solid waste around chemical and fertilizer industries. He has developed more than 25 IPRs and commercialized most of them through technology transfer to industries. His current involvement in the area of nano interventions in agriculture, micro encapsulation technology, developed for beneficial at fragile microorganism and seed coating technology are few which are making inroads to benefit mankind. Dr. Alok Adholeya is also the chairman of National Task Force of Department of Biotechnology, Ministry of Science and Technology, Government of India, on biological agent. He was conferred most prestigious award from Science and Technology, Ministry Product Process and Technology Award 2004 for his exemplary and only technology in the world of commercial production of mycorrhiza or next generation biofertilizers. Through his advancement, India being developing country provided technology to developed countries such as USA. His path-breaking work received Marico Foundation Award for addressing global problems of industrial solid waste remediation. Dr. Alok Adholeya currently leads the research and technology platform on mycorrhizal research, nanobiotechnology, micropropagation technology, community farming and livelihood with over 250 employees. Dr. Adholeya has chaired numerous advisory committees on agriculture and bioremediation, published over 170 research articles, guided 26 doctoral students and many master students, and has been an honorary professor at Deakin University, Australia since March 2010. He also worked as Director, Product and Technology Development at Microalgal Application, Oregon, USA, between 2014 to 2017, which is a subsidiary company of Sumitomo Chemicals. He currently heads National Center of Excellence in Agri Nanobiotechnology of Government of India, Department of Biotechnology. I would also like to tell all the participants that I was fortunate enough to be a student of Dr. Adholeya sir during my master training, and he was my master dissertation project guide, and I have learned a lot. And today is very fortunate day that I have invited sir, and sir has very graciously accepted our invitation to enlighten us with his wisdom and knowledge. Thank you, sir. Over to you to deliver your talk.
Thank you, Vikas, and thank you, organizers, for this opportunity. First of all, my sincere apologies for this delay because of the technological hiccups, but finally we are connected. I am hoping that all of you are able to hear me. In next 35 to 40 minutes, I will try to give you a bit of what we have been doing and what is potentially possible by showing you the power of microorganisms in various sectors, and particularly the biorefinery as well as the land reclamation, which is industrial overburdens. So these are two areas which I would be covering to discuss with you, and then definitely I am looking forward for interactions with you, since all of you are the faculty in different institutions. First of all, the TERI was established in 1994. It's a 100% research organization. We have presence in many other countries besides presence in India in different places, as you see on the screen. However, the important aspect is that the TERI is a unique organization, which is not-for-profit organization, non-government organization. And we have grown over a period of time fairly rapidly. We have 1,400 employees today, and we earn all our requirements through projects, consultancies, technology transfer, license fees, and so on and so forth. That means there is no check written to this organization. And this is something we say with pride, that we earn our own money and conduct research. The skill sets which are available with TERI are very many. The microbiology is a central theme which is a big strength of TERI. We have huge number of culture collections, one of the world's largest culture collection of mycorrhiza, but also a huge collection of bacterial strains, actinomycetes, and so on and so forth. We work in the nanotechnology area since 10 years and been able to develop some products which are going into market now. There are some technologies of manufacturing nanophosphorus, nano zinc and iron, nano sulfur, and these are again biologically synthesized processes. So they are different than physical or chemical processes. the very safe materials that we are producing. We work in rhizosphere biology, we work in therapeutic proteins, organic farming, culture, bioreactors. We have bioenergy area and the bioremediation that I'll be talking about, and also the biorefinery area, the plant molecular biology. The microalgal technology for bioremediation, as many of you must be aware, the algae captures CO2 from thermal power plants, and that is one emission that can be easily used by algal system. It's a very powerful organism in that sense, and convert that for energy production in the lipid manufacturing, as well as for several metabolites that it produces. Algae can also grow in wastewater, and that's where the lots of exploitation worldwide have been done. A number of different technologies have been developed. However, the important aspect is that selection of the specific algal strain for a specific problem is something that requires continuous progress. And I really encourage all of you faculties to try and see if you are interested and pursue this area. This is in a very interesting area. There is now even algal systems have been used for energy generation, electricity generation as well. And there are a number of different programs in Europe being conducted in this area. Now, the microalgal biofuel is something that we always hear. The important aspect is that the biofuel from biological systems are generally not economically sustainable. And therefore, there is a need to work in various different areas. For example, as you are fully aware, that when you get a crude oil, the crude oil undergoes several processes, which is called refining process. And refinery process produces a number of different products. And similarly, if the algal process is also dealt with the refinery process, then you produce number of products in future. And that's what I would be touching upon a little later. The important aspect is that India is quite fortunate, having an appropriate temperature across the board, except the Himalayan region. We have sewage water, which is huge, 62,000 million liter per day, and 7,500 kilometers of coastal line. That means a huge opportunity for producing algae in our country. Now, there are a number of products which, as I mentioned to you, can be produced from in the category of nutraceuticals, cosmeceuticals, therapeutic proteins, and pharmaceuticals as well. The fatty acid and sterols, which includes EPA, DHA, ALA, GLA, SDA, and several other OL compounds besides carotenoids, single cell proteins, and vitamins. Now, in the cosmeceutical area, moisturizers particularly, and the carotenoids, polysaccharides, antioxidant enzymes, micropore like amino acids, and skin whitening and healthcare like fucosandia and microalgal extract. These are some of the products which are drawn from algal system. Similarly, the therapeutic protein, the AB against anthrax virus, against herpes simplex virus, Phytex and simplex microalgal extracts are also used for tyrannus inhibitors. So there are a number of possibilities. The same algal system, same organism can produce multiple compounds. And that's what we've been talking about, to use microalgal system in a biorefinery process to produce biofuels, chemicals, food and feed, food additives, cosmetic and healthcare products, and pigments, carbohydrate, proteins, and lipids. So that's the power these microorganisms have. What we need to do is to deal with them. And in fact, there are a number of programs now in India which deals with them. The collections that we have done at TERI, of course, there are a huge number of collection of microalgal systems worldwide. We have collected some of these freshwater systems, including even a lake in Ladakh, but also collected from the coastal area. These are 733 isolate, fresh water is 317, and marine water is around 416. So these are number of isolates that we have. And what we have done the bioprospecting, we call it phycoprospecting, in order to find out whether they are producing lipids, they are producing beta carotenes, and then how they are producing and whether they are also suitable for the biofuel production. And huge amount of analysis has been done using different species, as you see on the screen, of microalgal species. Yeah.
Sir, so the screen is not visible, sir. If you could try sharing again, sir. Sorry.
Is it not available since beginning?
Yes, sir.
Oh, let me try and see if.
So now it is on, sir.
Okay. So you are seeing now my screen or what?
Still it is blank. Now it is appearing, sir. Phycoprospecting.
Yeah. It's there. Is it there now?
Yes, sir.
Okay. All right. So my sincere apologies for not able to show you earlier, but so what you're seeing on the screen are different species and the bioprospecting that we have done. The contributions that has been made by us in the improved cost economics in the biorefinery, we have produced not only the lipid on the higher concentration, but also a number of nutraceuticals like astaxanthin, lutein, and even beta carotenes in sufficient quantities, which makes economically attractive situation for the microalgal biofuel. The strain have been selected particularly, and as you would see, threefold enhancement has been done by employing rate limiting factors. And by this, of course, number of publications that have come up by one of my PhD student in this area. Now, we also have the outdoor production system, and now we have set up in Mumbai a major outdoor facility for production. This is something where the Center of Excellence program have come up in TERI. And in this program, we are able to optimize number of different products on one hand, as well as optimizing large scale production of these strains that we have selected. One of the Dunilila tertiaracta strain have been selected for this work. And 10,000 liter photobioreactor, as you see, has been established in Mumbai. The value chain conceptualization includes the EPA, the proteins, the lutein, astaxanthin, and keeping in mind the climate, productivity, capital cost, and manpower and drying steps of alga. Now, what we have also developed, a colleague of mine have developed the wet stage extraction of lipid so that there is no drying, because the drying takes and the dewatering takes huge cost. So to avoid that, the wet stage extraction has been developed. Now, as I was talking about in the beginning, the algal bioprocess and the refinery, as you would see on the screen now, there are a large number of products which have been developed by different species of algae. and they have been some of them are getting into commercial stage. So food, feed, fuel, waste valorization, mineral mining, and the nutrition and designer foods. So a number of different species, for example, Hematococcus, Nanochloropsis, and Dunaliella are the species which have been used for lipids, pigments, and biogas and biodiesel. Then there are species which are producing the minerals on one hand, the Cyclotella, the Spirogyra species have been used for pulp, fiber, and food and feed. The Chlorella spirulina, both these species have been used for the harvesting in-house, the SCPs and algal bioprocess. Now, so there are a number of those that have truly participated in the biorefinery process. Now, if you look at the circular economy point of view, the biomass, the food, feed, and then the whole thing comes back to the system, making it a circular economy by having provisioning, regulating, and cultural aspects, the aspects of revenue generation, aspects of meeting food and feed requirements, as well as the energy requirement. So the circular economy model can be implemented, it is feasible, and one need to definitely look at it. There's another very important aspect the whole world is talking about, the hydrogen. There is a possibility of biohydrogen production from this system, and that will be additional, I would say, milestone that would change the energy production possibilities from a material from the microorganism, that is microalgae. Waste, of course, there are a number of industrial waste, and the dairy industry have been using the microalgal systems for even cleaning their wastewater. But there is tremendous potential in several other sectors. Wastewater sludge, something that has been developed, and then even the biomass again is recycled and used for a number of different products. That completes my discussion with you about the microalgal system and the biorefinery that is a future. There is a tremendous amount of research inputs which are required at this stage, and I would encourage all of you to look at this as an opportunity of research contribution that you can make. Now, I will be talking about another area that is the industrial overburdens. As you know, many industries in our country and worldwide produce waste, solid waste, which remains on the ground. And there are a number of health hazards, there are a number of other poses to the mankind or the ecosystem. So we have been working with many industries in the past and developing technologies which are useful and making opportunity for industries to handle these in a manner which are safe on one hand, and also generate revenue which can take care of burden sites. So that the other aspect is that over a period of time, it also gives you revenue return. And such technologies are very useful. In this case, that we have been using no good earth or no chemicals. That means we are growing plants. We are using microorganisms, which we have selected. One of the central microorganisms that we have been using is Mycorrhizae. These are, I will explain to you if you do not know about these organisms a little later. We have also been selecting plant species which are suitable for the local environment, because we have been working worldwide and different agroclimatic regions in India as well. So selection of plant species is very important. We use organic compost, generally use the compost from the site itself, and then otherwise bring in compost from elsewhere and then do a reclamation work. To minimize and economize water use, we use drip irrigation system, and also in a manner in which the water use is minimized, because some of the sites we worked with were not having sweet water. So minimum water requirement, and at times mixing with seawater was also done, 70% seawater and sweet water in order to reclaim. So as you would know, mycorrhiza is a combination of root and fungi, which is a natural symbiosis which happens in nature. And they help in plant growth because these organisms have thread-like mycelia, hyphae. Those goes beyond several meter beyond of the root area. That means the area of absorption of nutrient, area of absorption of water, and mineralization is another function by which the nutrients are available to plant. So plant gets the raw materials, and then plant do photosynthesis, produce carbon. And since these organisms are not able to synthesize their own food, they draw very small amount of carbon from the plant as a friendship gesture. So these organisms are definitely very useful, and we have been exploiting them for agriculture, but we have also been exploiting them on such difficult sides of the industries. How they work, as I explained to you, they improve, they mobilize nutrients from soil, bring it to the plant system, convert non-available forms of nutrient to available form, and then the photosynthesis happens, the productivity enhancement happens, and the carbon is drawn. As you would see in the conventional plant system versus the plantations which receives mycorrhiza, the root biomass increases also because there is more carbon available, and therefore the root biomass increases. And that's how the network of mycorrhiza and the root is able to exploit maximum area of soil for minerals and so on and so forth. Number of benefits, as I mentioned to you, saving fertilizer, high soil temperature, they work very well from -10 to 60 degrees Celsius. These organisms are active, they do function very well, and they provide benefit to the plant system. For you, this is something which I wanted to show you, that we have worked on different solid waste from different industries. As you would see, we have worked on phosphogypsum. We are currently working with phosphogypsum with 3 industries. Zerofix is a zinc waste from the Hindustan Zinc Limited, which is also a waste that we have been now using for producing nanofertilizers, but also requiring such waste initially. The fly ash ponds, we have more than 40,000 hectares of our useful land under the fly ash, because we have used lots of coal for generating power in the past, and we are still doing it. And because of that, 40,000 hectares of land in India is under fly ash. This fly ash is an ash which is generated after the combustion of coal. And this is having large number of heavy metals and no growing capacity. And it's a very light material. It becomes airborne. It gets into bronchial system of humans and animals, and also causes several other difficulties because of the metal it contains. So we have worked tremendously on this area with several thermal power plants in India. We have worked with Tata Chemicals for many years, and we are still working with them since 20 years for their chloralkali sludge reclamation. We have done that in first few years of 60 acres of periphery of Tata Chemicals in Meetapur. And now we are dealing with their other waste reclamation and other waste valorization. We have worked in Qatar and Middle East for hypersaline desert soil. And we have also worked with the distillery effluent liquid waste versus as well as the solid waste from this distillery in Madhya Pradesh. This is Coromandel land, which a phosphogypsum pH was 4.4. rich in soluble salts. Electrical conductivity was 9.3. As you know, the electrical conductivity needs to be less than 1 for growing crops. So this vegetation was not supporting, this land was not supporting any vegetation. Of course, the erratic rainfall, but still you do not see any vegetation on this surface, and that was the biggest challenge. In 2013, we have taken first phase of the project. In 2016, as you would see, the entire area was reclaimed. And as you might remember, we don't use any good earth or chemical fertilizers to grow this vegetation. So we have used microorganisms, selected microorganisms for this reclamation activity. We have taken recently one more 11-acre patch of theirs that we are currently reclaiming. The physicochemical properties are, as you would see, and as I explained to you, the good amount of phosphorus and potassium, but good amount of sodium and magnesium salts as well. And that was posing a lot of problems. There was a lead, cadmium, chromium, and also in this. So that's something which also been addressed by phytoremediation activity. The irrigation water was also carrying, was an alkaline water, and the leachates were coming there. So that's also was the problem. And the challenge was that this pollution control board wanted that there should not be any leachate which should carry anything hazardous. And what we have done by this technology implementation confirmed that there is a ceasing or stoppage of leachates moving into other areas because they have been immobilized in the microorganisms or the plant system and without causing any toxicity. As you would see, with the plantation from the zero time, which is blue, and then after 20 months, as you would see, there is a reduction of heavy metals and increase of nutrients, and that is how the plant vegetation has started flourishing. We have used, as you would see, number of these isolates, which we have also isolated from sites, which we will be using in future. From the rhizosphere, because these isolates are now growing into the system, and these organisms can also be very useful in future for reclamation activities. This is Hindustan Zinc Limited, as you would see, a very difficult site to deal with, and we have conducted these plantations with microorganisms again. What we do generally is do microcosm experiments to select microorganisms from our germplasm bank. And once we have selected these microorganisms, we use them for reclamation activity. And generally, we are very much successful. As you would see that in late 2018, we have taken this photograph, and 2020, in 1 year's time, we had very good growth. This plantation is only 5 to 6 month old plantation. So it takes a while to develop the entire system, lay out the irrigation channels in order to use water efficiently, and with the difficulties, as you would see, the high amount of zinc and lead, which was a major problem at this site. As I mentioned to you, thermal power plants we have worked with the Badarpur Thermal Power Station. We have worked with Korba Super Thermal Power Station as well as the Vijayawada Thermal Power Station. And these power stations, we have used number of different interventions. At Badarpur, we have grown tree species along with floriculture plants. At Korba, we have grown timber trees, bamboo, as well as a number of other tree species, even casuarina on this site. And at Vijayawada, we have used medicinal plants, even aloe vera and the tree species in combination. There's always a combination because the challenge has been that we have to arrest, as you are seeing on the top here, the ash which becomes airborne very easily. And because of this problem, we have to have the vegetation which arrests this fly ash and allow and thrive these plants. Sorry?
Participant, please mute yourself.
Thank you. So we have also looked at how heavy metals have been reduced over a period of time. I must mention that all these demonstrations are still live, and we are managing them in order to develop a lot of data sets. And after 10 years of reclamation, we have studied the flora, fauna on the site, and also looked at how the heavy metals have reduced or changed over a period of time. And as you would see, the heavy metal reduction was significant on this site. The kind of biomass we are able to produce was huge. And as you would see, this is vetiver, which is a very good grass, very useful grass, used for making syrup, but also used for several other nutraceutical ingredients, the metabolites from this. More than 7 feet deep root have penetrated in this area, and as you would see, the plantation and under that the vetiver growing very well. This was after 3 years of vegetation on the site. So as you would see, the vegetation was thriving well. And this is a picture on the right side with red dots on the hyphae of mycorrhiza, was showing how the heavy metals, this is chromium, which is adsorbing on the mycorrhizal mycelium. So this is how the metals have been arrested by the microorganisms and the plants having no toxicity of these heavy metals and thriving very well. This is Tata Chemicals, which is alkali chlor sludge, very difficult waste material from the industry, and marshy land because it was on the sea level. And we have planted a number of local tree species by selecting microorganisms, suitable microorganisms, which can do symbiosis with these tree species. And this is something which we say with pride that the Tata Group of Industries have been showing this clip of Mithapur, known as Malara Green Cap project, in their corporate movie. So this is a great success, and because of this success, we have been working with Tata Chemicals since almost 20 years. This is another of their problem site. This was slopy land which we have reclaimed. These are the raisings of filling of different waste material, and now we are working on the waste material, another waste material with them. This is in Qatar, in Doha, Middle East, which is the hypersaline sandy soil, which is low-lying at the sea level. As you see, the electrical conductivity was 72, and the chlorides were 873, sulfates 175, because it's a sea level, so it's a sea water there. So reclamation, and there was no sweet water available. So we have used 30% of the sewage treatment plant water, wastewater, and the rest the seawater, and we reclaimed this site in 2009. Another industry which we worked in 2001 to 2003 using their brown liquor. This is a distillery which does not have the associated sugar mill. And so they buy molasses and produce alcohol, and they have a biggest problem of the brown liquor which comes out. And otherwise, the sugar industries use this material into composting. So here the problem was that they don't have any of that utility possibility. So we worked on reclamation activities and high transpiration rate species so that this wastewater can be utilized and then concentrated material can be treated. We have worked on the chromium extraction from the Kanpur area using a microorganism, which is a very useful microorganism that led us to actually move into nano area, realizing that these microorganisms are able to convert this chromium 3 and chromium 6 from the ternary sludge into nanoform, and then they are able to accumulate. As you would see on the number 3 picture on the screen, this is a green color. This is actually a fungal strain which has extracted 40% of their body weight chromium into it, and that's why the biomass has become green. The organism is still alive, it's not dead. So the amount of chromium it has been able to absorb and accumulate within the biomass was phenomenal. And this particular system was used. We published multiple papers from this work as well. Of course, we have received multiple awards for these industrial overburden activities. We published a number of papers from this activity, number of projects that we have conducted with industries, I will go quickly. And we have worked with Ministry of Science and Technology and the industries for these activities. These are publications of ours and land reclamation. Thank you very much. I appreciate your patience. Because I think you are on mute.
Because you're mute. Yes, sir. Sorry. Is it okay now? Yeah.
Yeah.
Yeah. Thank you so much. I was not able to unmute myself. Thanks a lot, sir. Such a nice and wonderful presentation, and we got to know so many technologies about that TERI is working on, and we are very much aware about the TERI's contribution into this domain, and already these technologies are being well famed. And today we are fortunate enough that we have witnessed presenting one of the great leader into this domain to us. And now I would like to invite participant. If anyone would like to ask a question, so the house is open now. Anyone like to interact with us, sir? They may ask the question of their desire. Any participant, please? Please mute your mic first, and you want to ask any question, you please ask. Sir, there's one question from my side.
Go ahead.
Having seen such critical issues in degraded land, and there's a great application of mycorrhizal technology. So can we utilize this as a whole source of heavy metal remediation?
Yes. What we have now been doing is that the mycorrhizas are very good in heavy metal accumulation. And since they do not hound them, they are able to accumulate huge quantities. So what we are now doing is extracting those pure metals from mycorrhiza, and they become a product. Okay. So they are not only accumulating good quantities, but also we can draw a product, because the pure metals are a very costly commodity, and we can separate number of pure metals from the mycorrhizal system.
Okay, so basically I have asked this question because we are already doing some work in our lab in our college, and there we are working on a hydroponic system using some plant system to extract heavy metal from industrial waste. Okay. So I was wondering if this particular microorganism could be utilized in those hydroponic system.
The aeroponics, it works very well. In the hydroponic system, it might not work very well because these fungi requires better aeration.
Yes.
And the submerged condition, fungi don't like that very much. So yes, aeroponic system, it will work very well.
So what if we can devise some system where we can provide some rheology of the broth that fungus is going to generate? So if the rheology is being addressed, so can we maintain uniform heat and mass transfer inside the reactor system of hydroponics?
Sure.
So will that be kind of technology so that we can propose some novel method in this domain?
Absolutely. You need to optimize a bit on combining the systems, but definitely you can do that.
So what are the critical optimization challenges into this domain? I mean, apart from rheology.
The critical optimization have to be that, first of all, I need to know what is your objective of growing hydroponics. If you're growing hydroponics for treating the wastewater, and that is the only intention, then and you're growing plant species which are hyper accumulating, I believe, that you're using. So
that
if
is a sorghum plant we have tested into our lab.
Right. So you must know that the mycorrhizal fungi do not colonize or make symbiosis with brassicas. And there are a number of different brassica species, which is hyper accumulating species. So you need to find other plant species which can have symbiosis and allow mycorrhiza to grow extensively beyond the rhizosphere. So that's one optimization aspect. The second optimization, of course, is that you need to make a selection, which is the isolate, which is suitable in the hydroponic situations. And if you are creating dry cycles, then that should be optimally suitable for given conditions. The third aspect, of course, and then the selection is not only growth, but also hyper accumulation properties. So you need to make some selections of mycorrhizal strains for that particular purpose.
Yes, sir. There is one more question from participant side, sir. Is there any possibility to convert poultry and fish waste to biocompost and use them to grow medicinal plant accumulating pollutants?
Yeah, there are a number of technologies which are available. Fish waste is being composted very well. You can do aerobic composting. You can do even the reactor-based composting. Fish waste is a very good waste. It has lots of nutrients. And growing plants in the composted material is very easy. So you can do aerobic composting. What is the another waste that he has talked about? Fish and poultry.
Fish and poultry, sir.
Both waste have been composted very well. So yes, there is a total possibility of using them as a compost eventually. Very powerful compost.
Okay. One more question is there, sir. Sir, can we use mycorrhiza for remediation of heavy metal from groundwater?
Yeah, sure, definitely. What we need to do is to bring water as an irrigation material, and then the mycorrhizas can accumulate heavy metal from there.
Okay, so I mean the work that we are going to have in our lab. May I request you to have your suggestion and guidance in future too? That may I contact you in future so that I can get guidance from yourself.
Definitely, most welcome.
Thank you, sir. And I hope there is no other questions from participant side. I would like to announce once more that if anyone want to contact or interact with the sir, they may interact right now or afterward this session. And it was really very wonderful session, sir. And I would like to thank you for accepting my invitation to join us in this event.
To interact, and there are a number of so many 66 participants I can see.
Yes, sir.
I would like to thank them for being available here.
A few of them have not able to join, sir. However, there were 98 or 99 participants. Some of them has not joined because of this technical issues.
I know that was a very difficult.
So thanks a lot, sir. Thanks for giving us such a brief and wonderful insight, sir. Thanks a lot.
I appreciate it. Thank you. Thank you.
Thank you, sir. Yes, Abhinav, sir. Okay, participant, I guess we are done for the day. And sorry for this technical glitch today. There are several issues into online session, but somehow we could manage. And thanks a lot for your support and kind gesture that you have joined at this portal. Tomorrow onwards, we'll be having same portal, the Microsoft Team app, since we are having that licensed one, and still we don't have Cisco in this mode. So we'll be carrying on our session of the day on Microsoft Team app only. Thanks a lot. Hello.